right. Today I am going to explain about amphibolic pathway. So, in this amphibolic pathway, it is the amphibolic pathway. It is biochemical pathway that includes both catabolic and anabolic reactions. So, this is the definition of amphibolic pathway. So, in this amphibolic pathway, it is a biochemical pathway that includes both catabolic and anabolic reactions. So, what is the catabolic reactions? Generally, in previous classes, I explained about catabolic processes like glycolysis, link reaction, Krebs cycle. So, in this catabolic, what is the catabolic reactions means that catabolism is the one type of metabolism. The catabolism and anabolism occurs during the cellular respiration. Here catabolism and anabolism occurs during the cellular respiration. During the cellular respiration, the respiratory substrates are oxidized into the simpler compounds. Okay, so that is the cellular respiration. Right. Next, what is the definition of the catabolism? Catabolism. So, in this catabolism, breakdown of respiratory substrates like carbohydrates, fats and proteins. So, in this catabolism, the breakdown of the organic food substances is known as catabolism. So, during the catabolism, the breakdown of the carbohydrates, breakdown of the fats, breakdown of proteins. Okay, so this is the definition of the catabolism. So, in this catabolism, the breakdown of the carbohydrates are nothing but that is example for the catabolism, glycolysis. Glycolysis is the best example for the catabolic reactions. So, next, what is the definition of the anabolism? Anabolism is the biosynthesis phase of the metabolism. During the anabolism, biosynthesis of organic food substances. Okay? So, here during the anabolic reactions, synthesis of newly synthesized organic food substances by the anabolism. So, what is the example for the anabolism? The best example for the anabolism, Krebs cycle. Krebs cycle. So, these are the basic concept about the amphibolic pathway. So, in this amphibolic pathway, it is a biochemical pathway that includes both the catabolic and anabolic reactions. So, this is the amphibolic pathway. So, next, in this amphibolic pathway, different types of respiratory substrates are participate in the amphibolic pathway. So, what are the respiratory substrates? Here, this is the respiratory substrates. In this respiratory substrates, basically 3 types are there. Basically, 3 types of respiratory substrates. The first one is fats, second one is carbohydrates, third one is proteins. So, these are 3 types of respiratory substrates are oxidation, undergo the oxidation process, then break down into the simpler molecules. Okay, na? So, that is the respiratory substrates. Now, here during the oxidative catabolism of carbohydrates. So, the carbohydrates are complex substances. These complex substances of the carbohydrates are converted into the simpler simpler molecule that is called glucose. So, these glucose is undergo that is oxidation process, then to form the here glyceraldehyde 3-phosphate. In between some reactions are there in glycolysis process. We can remember once glycolysis process. Here glucose is converted into the glucose 6-phosphate. Glucose 6-phosphate is converted into the fructose 6-phosphate. Fructose 6-phosphate is converted into the fructose 1,6-bisphosphate. That fructose 1,6-bisphosphate is a breakdown into the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So this glyceraldehyde 3-phosphate is again converted into the pyruvic acid. So this is the end product of the glycolysis. The pyruvic acid is end product of the glycolysis. Then the pyruvic acid is again converted into the acetyl-CoA by the link reaction. Next, acetyl-CoA is again breakdown into the acetyl-CoA is again converted into the oxaloacetic acid by through the Krebs cycle. So this is the complete oxidation of the carbohydrates. So what is the amphibolic pathway? Here amphibolic pathway includes catabolic and anabolic reactions. Here carbohydrate oxidative catabolism of carbohydrates, then to form the pyruvic acid to acetyl-CoA is again entering into the Krebs cycle. So this is a carbohydrate catabolism. So next, fats. This is the fats. These fats are oxidative catabolism of fats then converted into the glycerol and fatty acids. The fats are undergo oxidative decarboxylation then to form the fatty acids and glycerol. These are fatty acids. Here glycerol. So this is the glycerol. This glycerol is converted into the dihydroxyacetone phosphate. This is dihydroxyacetone phosphate. Again, this dihydroxyacetone phosphate is again converted into the glyceraldehyde 3-phosphate. Then the glycerol 3-phosphate is again converted into the pyruvic acid, then entering into the Krebs cycle. So next, end product of the oxidation of the fats, the fatty acids are formed. These fatty acids are undergo, that is by the beta oxidation process, the fatty acids are converted into the acetyl-CoA. Again, this acetyl-CoA is again entering into the Krebs cycle. So here fatty acid, for example, famitic acid. Famitic acid is example for the fatty acid. So it is contains 16 carbons compound. So when the famitic acid is undergo oxidation process, then to form the 8 acetyl-CoA molecules are formed. These 8 acetyl-CoA molecules are again entering into the 8 cycles of the Krebs cycle. So this is the beta oxidation process. During the beta oxidation process, fatty acids are converted into the acetyl-CoA. Acetyl-CoA is again entering into the Krebs cycle. So this is the breakdown of the fats. So next, the third respiratory substrate, that is called proteins. During the oxidative catabolism of proteins, then to form the amino acids. So this is the amino acids. So when the proteins are break down into the amino acids by the protease enzymes. So these protease enzymes break down the proteins, then converted into the amino acids. So after the formation of the amino acid, after the amino acids are undergo deamination process, then to form the again pyruvic acid is formed. So this is the pyruvic acid. Again the amino acids are undergo deamination, then to form the pyruvic acid. Again pyruvic acid is again converted into the acetyl-CoA. This acetyl-CoA is again entering into the Krebs cycle. So this is the breakdown of the fats, breakdown of the proteins. So these are 3 types of respiratory substrates are oxidative catabolism of fats, oxidative catabolism of carbohydrates, and oxidative catabolism of proteins. So these respiratory substrates are undergo that is oxidative catabolism. Then that end products are again entering into the glycolysis and Krebs cycle. So this is the catabolic reactions. So next, what is the anabolic reactions in the Krebs cycle? So see, this is a Krebs cycle. Once we can remember the Krebs cycle in previous videos, that is in this Krebs cycle, the acetyl CoA is condensed with the oxaloacetic acid then to form the citrate. Then the citrate is converted into the alpha ketoglutaric acid. Then alpha ketoglutaric acid is converted into the succinyl CoA. Succinyl CoA to succinic acid. Succinic acid to the malic acid. Malic acid to the fumaric acid. Then the fumaric acid is again converted into the oxaloacetic acid. So in this, during the glycolysis, some intermediate products are formed. These intermediate products are mainly involved in the precursor for the synthesis of the new organic compounds. Okay. So for example, the first stable product in the Krebs cycle, citrate. This citrate is the main precursor for the synthesis of the fatty acids and sterols. The citrate is intermediate product of the Krebs cycle. The citrate is the main precursor for the synthesis of the fatty acid and sterols. So next, alpha ketoglutaric acid. It is the most important intermediate product during the Krebs cycle. Alpha ketoglutaric acid is the main precursor for the synthesis of the glutamate amino acid. This glutamate amino acid is the main precursor for the synthesis of arginine, synthesis of proline, synthesis of the glutamine. So these three amino acids are synthesized from the glutamate. This glutamate is synthesized from the alpha ketoglutarate. So this is the alpha ketoglutarate is the main precursor for the synthesis of the three amino acids, that is glutamine, proline, arginine. So next important concept here, the alpha ketoglutarate is the main precursor for the synthesis of the purines. Okay, the alpha ketoglutaric acid is converted into this is the main precursor for the glutamate. This glutamate is the main precursor for synthesis of the purines. What are the purines? The purines are two types, adenine and guanine. So adenine and guanine means the purines are synthesized from the glutamate. Glutamate is synthesized from the alpha ketoglutaric acid. So this is the one of the intermediate product in the Krebs cycle. So next, the most important concept here, that is the succinyl CoA. That succinyl CoA is the main precursor for the synthesis of the porphyrin ring of the heme groove. Porphyrin ring of the heme group for synthesis of the hemoglobin and the synthesis of chlorophyll. So this is the main concept. The succinyl CoA is the intermediate product in the Krebs cycle. The succinyl CoA is the main precursor for the synthesis of the porphyrin ring of the heme group. So this is the succinyl CoA. So next intermediate product, that is the oxaloacetic acid. This is the oxaloacetic acid. This oxaloacetic acid is converted into the phosphoenol pyruvic acid. This phosphoenol pyruvic acid is the main precursor for the synthesis of the serine, synthesis of the glycine, synthesis of the phenylalanine, synthesis of the tyrosine. So these tyrosine and synthesis of the tryptophan. So these are 5 amino acids. These 5 amino acids are synthesized from the phosphoenol pyruvic acid. This phosphoenol pyruvic acid is synthesized from that is oxaloacetic acid. So this oxaloacetic acid is the main intermediate product in the Krebs cycle. So from the phosphoenol pyruvic acid, then synthesis of the serine, glycine, phenylalanine, tyrosine, tryptophan. These these 5, that is amino acids are synthesized from the phosphoenol pyruvic acid. So next, this is the end product, the end intermediate product in the Krebs cycle, the oxaloacetic acid. This is the oxaloacetic acid is the main precursor for the aspartic acid and aspargine. So these are the main amino acids for synthesized from the oxaloacetic acid. This oxaloacetic acid is the last intermediate product in the Krebs cycle. The oxaloacetic acid is the main precursor for the aspartic acid and aspargine. So these 2 amino acids are main precursor for the synthesis of the pyrimidines. So what are the pyrimidines? Cytosine and cytosine and pyrimidines. What are the examples of the pyrimidines? Cytosine and thiamine and uracil. So these 3 types of nitrogenous bases are present in the pyrimidines. So here the oxaloacetic acid is the main precursor for the aspartate and aspargine. These two amino acids are main precursor for the pyrimidines. These pyrimidines are for synthesis of the cytosine, thiamine, uracil. So these are pyrimidines. So here succinyl CoA is the main precursor for the synthesis of the porphyrin ring. Here alpha ketogluteric acid is the main precursor for the glutamate. The glutamate is the main precursor for the purines. The purines are adenine and guanine. Here pyrimidines are synthesized from the aspartate and aspargine. These 2 are synthesized from the oxaloacetic acid. These oxaloacetic acid is the main precursor for the synthesis of the pyrimidines. That is, the pyrimidines are cytosine, thiamine and uracil. So this is the anabolic reactions. Anabolic pathway is nothing but biosynthesis of the phase of the metabolism. Biosynthesis phase of the metabolism means synthesize the new compounds, new molecules from the Krebs cycle. So that's why that anabolic reaction is the best example for the Krebs cycle. During the intermediate products of the Krebs cycle, different types of the amino acids are formed during the amphibolic pathway. So this is the complete explanation about the amphibolic pathway. Okay, thank you.